Hey everyone. Today we are talking about different eukaryotic organisms in lesson 5. So in previous lessons, we've discussed prokaryotes a lot and different techniques that are used in the lab, but now we're going to move on to talking about something that is somehow astoundingly more complex, which is eukaryotic life. Now, to give a quick review, prokaryotes lack nucleuses and are always unicellular. Eukaryotes have nucleuses and are often multicellular, but sometimes they are unicellular. So that's something that we have to be aware of when we're differentiating between prokaryotes and eukaryotes. Oftentimes, even myself, I mess up and I say, eukaryotes are multicellular organisms, whereas sometimes they can be unicellular. So that is a commonality between some eukaryotes and some prokaryotes, that they are unicellular. And again, that kind of falls into that same rhetoric that I've been repeating, where you have buckets, and all those buckets will have some sort of overlap. So when we're looking at things, just keep in mind that the definitions can at times throw you for a loop, as they do me very often. So when we're talking about eukaryotes in the microbiology world, we're going to be talking about pretty much four main categories. And again, with buckets and categories, there's always some weird overlap, but the categories are protozoa, fungi, hemolymph, and algae. So those are the main things we're going to be talking about here today. Now, remember, obviously, I'm a eukaryote, you're a eukaryote, but that is on a obviously macro scale. So in micro, we're looking at those four things. And to break this up, we're going to talk about eukaryotic parasites first. So this is going to be your protozoa, which are unicellular, and hemolymph, which are multicellular. So protozoa are actually what are called animal-like microbes. That just means that they can move around their environment and consume things like a animal hunting prey or a herbivore going up to leaves and chomping them. So they have cilia, flagella, or some sort of other way to be motile in their environment. So they're going to be moving around their environment a lot. And some examples of this are Plasmodium, which is actually malaria. So that is a protozoa, a unicellular organism. And that is going to be, of course, transplanted from one person to another person through the host organism, which is a mosquito. When we're talking about disease, you might think of yourself as the host, right? Because it's in you, but there's always going to be a host reservoir. And that host reservoir is where the disease is doing most of its living for a lot of the time. For something like malaria, that's going to be in mosquitoes. So that's the reservoir for malaria is going to be the mosquitoes. And then, of course, it gets transferred to people. And then obviously, we have a detriment to ourselves, but we're not the reservoir necessarily for malaria. Similarly, there's a lot of diseases that are carried by bats, and bats act as the reservoir. Bats are really interesting because when they're in flight, their internal temperature increases a lot. And with that internal temperature increase, they're actually able to sort of tamp down infections. So they're really good carriers and good reservoirs for infectious disease that then will spread and spill over into other animals and people. So when we're looking at something like Ebola, for instance, and that's a virus, so that's outside of this discussion of eukaryotic microorganisms, but Ebola and other diseases that are transferred by bats use the bats as the reservoir, but when they get into humans or other animals, obviously cause very, very severe disease. And that's something that all parasitic or viruses or pathogenic microbes will have in common is usually there's some place or organism that they're in. So parasites specifically have to have an organism that they typically live in, and then they spill over into something else. And that brings me to parasitic worms, helminths. So helminths are multicellular parasitic worms. They have what we'll call organs and tissues and specialized structures that allow them to attach and get into the body. Some examples of this are tapeworms and pinworms. And the reason these fall into the same overall category of parasites is they're dependent on the host for nutrients and survival. With worms, a lot of times that host is actually going to be humans, so it spreads between humans to humans. If you have a tapeworm, that is going to spread from an animal to a human, or a human to another human, or a human to an animal, typically. So parasites dependent on the host for nutrients and survival. And then they typically reproduce either in the host or in whatever organism that they're in that maybe isn't that reservoir, but is acting as the host at the time. So protozoa have two different types of reproduction. There's asexual mitosis and sexual meiosis stages. So they have these very complex life cycles. And once they're into the host, they will start undergoing those life cycles. So, for instance, Plasmodium and malaria, they only undergo a certain portion of their life cycle in the mosquito, and then they need a human or animal host to undergo additional parts of that life cycle. Hemolytes reproduce sexually, so either separate sexes or both sexes in one worm. So that's a weird thing to think about. So you can have two worms that have each of the different sexes, or one worm that comprises both, and they're going to reproduce with both sexes, but it can either be as one worm or two worms. So that's a little confusing, probably, but it's just life is very weird, and fitting things into boxes is never going to be foolproof. So just be aware that they can produce either as one worm or multiple worms, but they have to have both those separate sexes involved. Kind of confusing. When they do reproduce, they have intermediate hosts and definitive hosts. So, intermediate hosts occur during the larval stages, and definitive hosts is when they get to the adult stage. And this is when we're talking about reservoirs. Sometimes those are intermediate hosts where they are just in their larval stages, and then once they infect a human, for instance, they become we're the definitive host, so they become the adult worm, and then they will reproduce and make more larvae, and then those larvae will then go on and infect more people. So a takeaway is that understanding these differences in life cycle will help in the identification, the treatment, the study of these different parasitic eukaryotes. So obviously protozoa and these helminths are very easy to tell apart. One is a large worm, multicellular, and the other is a small single cellular, like a prokaryote might be. So we have those easy differences. When we're talking about these, the interesting thing is they're typically transmitted through contaminated food and water if it's not going to be through a vector like a tick, a fly, a mosquito. So this is, if you've ever traveled abroad, the advice might be don't drink the water out of the tap in certain places and never eat food that is not thoroughly cooked. And this is the reason, because a lot of these things they transfer through the eating of something that's infected or just will be in the water. There's a lot of videos online of parasites in stagnant water, and it is super creepy to look at. If you ever drink that water, you're definitely getting infected. So don't drink bad water ever. It's bad unless you really need to, just try and avoid it. So then we're going to talk about fungi after that, or fungi, however you want to say it. They have a lot of unique features because they're highly complex. They fill a lot of different niches, and they are very different from bacteria or archaea, but fill, again, a lot of those same niches and have this benefit of being multicellular. So, one of the ways they're different from bacteria is they have chitinous cell walls in a lot of cases. So this makes them really, really robust for surviving environments. So a couple of categories of fungi that are common. One, of course, is yeast. Yeast is unicellular. Remember that eukaryotes generally multicellular, but can be unicellular, and they produce by budding. So these yeast produce by budding. They bud off little bits, and then those bits grow into more yeast. So you have this budding, rapid turnover. So yeast can grow very, very fast. And that's why they're used in things like fermentation and used in, baking bread, producing beer, whatever it is. That is one of the great things about yeast is they do reproduce very fast from this budding. Another category is molds. Molds are multicellular, and they have these uh, filminous hyphae, so kind of can reach out and discover what's around them really easily, and they reproduce with spores. So we talked about spores a little bit in the lab safety of, hey, spores are really hard to kill. They're a huge pain for us for safety, but they're great for the molds because they'll bud, they'll have these spores, they can be carried on the wind, and they can get a bunch of places really easily. The problem is if those spores are infectious, then that becomes a huge issue because not only are they hard to remove from glassware in the lab, they're also incredibly hard to deal with in any sort of environment. Then there is dimorphic fungi. So they can exist as a yeast state or a mold state depending on the conditions. So yeast states usually is going to be when there's an abundance and they need to replicate really fast. And then the mold state is when there is less abundance and they'll kind of come together in these mats. But of course, that's going to depend on which fungi it is, what their environment is, if there's differences in the life cycle of them. So just know that there's yeast, there's molds, and there's these weird fungi that are a mix of two that can kind of swap between it and do whatever is needed to be done based on the environmental factors. Of course, a lot of these can be pathogenic. A lot of times it's opportunistic pathogenesis. So remember, opportunistic means that it's not normally going to be pathogenic, but it can be if all of your immune system's tamped down, you don't have the right microbiome or something like that, or you're on some sort of treatment that just is going to make it so you have a tamped down immune system especially in a certain part of your body. So I have asthma, so I take an asthma inhaler. You have to rinse your mouth out very thoroughly, or you could get what they call thrush, and that's a yeast infection of your mouth. The reason for that is the steroids are anti-inflammatory. They really tamp down the entire immune system in your lungs. That's the point of it. But it also happens in your mouth if you don't clean your mouth out. And then, of course, you're eating, your mouth is dirty all the time. It's full of yeast just from the environment and what you're eating and everything else. So you can get this uh, candida. It's not candida, it's candida infection, which is thrush in your mouth. Similarly, aspergillus is a type of mold that lets off spores, and the spores can be infectious. So we can get those opportunistic infections if, again, our immune systems aren't functioning properly or we're on some sort of drug, or even if we were just infected. So a lot of times people will get sick and then you'll fight off that sickness. It's a week-long flu, let's say, and then all of a sudden you wake up and you have some other infection that's completely unrelated, and then you're sick for another week because you were so worn out from that first infection. So another example of a pathogenic mold slash yeast is going to be athlete's foot. So athlete's foot is a mold. It's a huge pain if you get it. You have to be on drugs. Sometimes they're topical, sometimes they're oral, and then it, can take a long time for that to actually fully go away. In Arizona, especially, we have coccidioides, which is valley fever. So, a lot of people who are born here, you get valley fever when you're young. It's not too bad. You clear it really well. But if you're older and you move to Arizona or the Southwest in general and you get valley fever, it can be a debilitating disease where you have to go to the hospital. Sometimes people are in the ICU with it, and it's often not identified because of the way it presents. The reason all of these fungal infections are really hard to get rid of is because fungi, being eukaryotic, are a lot like us. So we can use antibiotics and get, a little bit of stomach upset, maybe some sun sensitivity, things like that from the antibiotics. Because bacteria are so different, it's really good at killing them, really good at letting us live our lives as normal. Same with parasites. Parasites are in this very specific space where they're doing very specific parasite related things, and it's easy to disrupt those life cycles because they're very different from eukaryotic cell life cycles. Molds and fungi and yeast, on the other hand, they're very similar to our cells. So on a cellular level, we're eukaryotic, very similar life cycles, and it's very hard to treat fungi without getting the person very, very sick from that treatment. And that's why a lot of things like the athlete's foot treatments, they're topical. You try and deal with it topically so you don't have to take a pill. If you have to take the pill that's an antifungal, it comes with a lot of potential side effects. So that's something to keep in mind. I think fungi are absolutely amazing. They're super interesting. They're very weird in the whole spectrum of life. They kind of sit in this place where they continually defy our classifications of them. So, one of the interesting things is I believe it is the biggest organism on Earth is a multicellular fungi that has spread out through an entire forest. So it's multiple acres of fungi underground, interwoven with the roots of these different trees. And it's beneficial for the trees, it's beneficial for the fungi. And they've taken samples, and in taking those samples, they've shown, wow, this is actually the same fungi. And we can tell because of the genetics. And it is, of course, that you have different diversity from one part to the other, just like you'll have differences between if we take, a muscle cell or a skin cell or a nerve and sample those, but the overall genetics is the same. So that is just completely mind-boggling that something so non-complex when you look at it in a petri dish could be so astoundingly complex in the environment. Similarly, algae fit some very interesting niches. So algae usually they're not going to be parasitic at all, but they're very important for us to understand. And the reason they're very important is not just for environmental science and understanding, the food webs in the ocean, but also because while they're not pathogenic, a lot of algae will produce toxins. And when they produce those toxins, the toxins can actually accumulate and they will accumulate in seafood and then things, of course, like birds eat the fish, something eats the bird. Ultimately, we have a very strict very regulated food sourcing system. But if you're out hunting and you hunt duck, let's say, and you eat that duck, you actually might get poisoned from the toxins that ultimately come from the algae growing in that lake because there's some sort of disruption happening in that lake. There was a case where they couldn't figure out why a lot of bald eagles, which are a protected obviously a protected species were dying around some lakes, and they traced it to the algae having an interaction with a plant that when the plant was subjected to a pesticide, it would produce certain toxins because the microbes that were a pest on the on the plant were kind of tamping things down in the plant, keeping it in an equilibrium. And once that equilibrium was disrupted, it produced toxins. The algae got the toxins, and then it moved up the food chain, ultimately through the fish to these eagles that were then dying. So it's this interesting interaction where when things get disrupted, oftentimes due to human intervention in some way where we see a positive, it has repercussions we weren't really thinking about. And one of the things with algae, especially, is this toxic buildup that happens over time and can especially accumulate in seafood and lead to poisoning of humans. So, very interesting. They're also, of course, interesting because they're one of the primary producers. Remember, we have producers and then things that eat producers. So the producers take in sunlight or other innate nutrients in the environment and then produce life and then replicate, and other things come along and eat them. So they're at the base of this food chain that moves up and up and up, and that makes them really interesting to study, of course. Now, some clear takeaways from this is eukaryotic microbes are incredibly diverse. They can be single-celled like protozoa, they can be these large complex parasites like worms, and knowing some generalities about them is very, very helpful, like their general structure, their general life cycle, and that all informs their pathogenic potential, how they cause disease, where they cause disease, and where we can come in and treat those diseases effectively. And you'll notice that a lot of these treatments aren't at the person level. They're actually going to be in the environment. They're going to be things like thoroughly cooking meats, screening the water for parasites, screening the water for, buildup of these algae that are producing toxins, things like that. So it really crosses over into more environmental monitoring and regulatory safety checks that we already have in place being adhered to so we don't get things like a bunch of people getting sick from things like parasitic worms in poultry or parasitic worms in just general meats like pork. Pork, for whatever reason, a lot of parasites live in pork, and that's why pork actually has a higher internal cooking temperature advisory than some other meats. So it's one of those things to keep in mind. Of course, there's things like different parasites that live in fish. So if you ever had sushi, there's sushi grade fish, and that's fish that has been frozen to such a degree that it kills all of the parasites that would be in the sushi, because parasites being complex and multicellular will actually die from deep freezing. So they're killed that way, and then they're fine. But if you don't get sushi grade fish, it's really important you cook it because it might have parasites in it. So again, super complex, incredibly diverse organisms that we're talking about when we're talking about eukaryotic microbiology, but there's some generalities that you can really key in on and use that as the basis for next steps in study. So, of course, the lesson details are going to be in your book, in the lesson write-up itself, and in the lab. And I wish you the best of luck on this lesson. As always, if you have any questions, please let me know, and I'll do the best I can to clear up any of these extreme complexities of the eukaryotic microbiology world. Have a great day.